Good morning, everybody. As President Wheeler mentioned, I'm Jeff Dirk, Dean of the Case Western Reserve University School of Engineering, and one of the co-chairs of this year's meeting. The 2014 EMBC meeting represents the 36th Annual International Conference of the IEEE EMBS. Under the overall theme of discovering, innovating, and engineering the future of biomedicine, the meeting covers a wide range of topics, from cutting-edge biomedical and healthcare technology research and development to the clinical applications in biomedical education. In addition to the keynote lectures by academic and industrial leaders, the meeting will also feature mini symposia, invited sessions, oral and poster sessions, educational sessions for students and young professionals, and exhibitions in over 170 sessions formed from almost 3,000 submissions. We hope that you have the opportunity to attend many of these exciting presentations and have stimulating discussions with your colleagues from around the world, as well as enjoy the beautiful city of Chicago. As part of the Keynote lecture program, it's my distinct honor to introduce you to perhaps the most accomplished leader and most respected pioneer in biomedical engineering histories, Dr. Leroy R. Hood. Dr. Hood's outstanding contributions have had a resounding effect on the advancement of science since the 1960s. Throughout his career, he's adhered to the advice of his mentor, Dr. William J. Dreyer. Dr. Dreyer said to him, "If you want to practice biology, do it at the leading edge. And if you want to be at the leading edge, invent new tools for deciphering biomedical and biological information." 100 years after the invention of the light bulb, the phonograph, the carbon microphone, and also X-ray fluoroscopy, American Thomas Edison is hailed as one of the greatest inventors of all time. Similar descriptions will be made of Dr. Hood. Dr. Hood was involved in the development of 5 instruments critical for contemporary biology, namely automated DNA sequencers, DNA synthesizers, protein sequencers, peptide synthesizers, and an inkjet printer for constructing DNA arrays. These instruments launched and opened the door to high-throughput biological data and the era of big data in biology and medicine that we all capitalize on today. He helped pioneer the Human Genome Program, making possible the automated DNA sequencer. Under Dr. Hood's direction, the Human Genome Center sequenced portions of human chromosomes 14 and 15. In 1992, Hood created the first cross-disciplinary biology department, molecular biotechnology, at the University of Washington. In 2000, he left UW and co-founded the Institute of Systems Biology, the first of its kind. He has pioneered systems medicine for years since ISB's founding. Dr. Hood has made many seminal contributions in the fields of immunology, neurobiology, and biotechnology, and most recently has been a leader in the development of systems biology, its applications to cancer, neurodegenerative disease, and the linkage of systems biology to personalized medicine. These are all words that we use commonly today in EMBC. Think about these accomplishments and their impact on this week's meeting. What would we be sharing this week had these accomplishments not been achieved? Hood is now pioneering new approaches to P4 medicine. Those 4 P's stand for predictive, preventive, personalized, and participatory. And most recently has embarked on creating a P4 pilot project on over 100,000 well individuals that is transforming healthcare. They're well individuals now, and as they age and as disease occurs, we will have data on them to understand what are the predictors of disease and how to treat them. In addition to his groundbreaking research, he is one of only 15 individuals elected to all 3 national academies: the National Academy of Science, the National Academy of Engineering, and the Institute of Medicine. Hood has founded or co-founded 15 different biotechnology companies, including Amgen, Applied Biosystems, Rosetta, Darwin, and Integrated Diagnostics, among others. He has published over 750 papers. He's received 36 patents, 17 honorary degrees, and more than 100 awards and honors, including, as you saw this past week, the IEEE's Medal for Innovations in Healthcare Technology, the highest honor the IEEE can bestow on an individual or team for extraordinary achievement in biomedical engineering. Please join in welcoming to the podium Dr. Leroy Hood for the keynote address for the 2014 EMBC meeting.
Well, thank you very much for that gracious introduction. What I'd like to do is give you a sense of what systems science is and how its application to disease has led to something we call both systems medicine and P4 medicine. But at the end, I'll talk about a project that I think absolutely will transform contemporary healthcare, both in the US and abroad. So, with that, let me say a part of my philosophy about creating companies has always been this feeling that we have a responsibility as an academic to transfer our knowledge to society. So I just want to say I'll be talking about the last 3 companies that I did found in the last few years, and I think these will be among the most transformative companies in this new approach to healthcare. So you'll get a sense of what those are all about. When I started at Caltech in 1970 as a young assistant professor, I was really struck by the enormous complexity of biology. I was a molecular immunologist at that time, and I realized that although we could do some very nice molecular work and deciphering theories of antibody diversity, we really had no clue about the most fundamental aspects of the immune system. How does the immune response work? How does tolerance work? How does autoimmunity come about? And in the context of having a really fascinating series of conversations with Max Delbruck, a Nobel Prize winner in the early days of molecular biology, I really started thinking about systems approaches, although in those days we didn't really have the technologies, nor did we have the strategies for thinking about how to deal with complexity. And in thinking about the complexity, it's clear why it arises. It is a consequence of this process of Darwinian evolution itself, random and chaotic, which is only interested in the solution to problems. And it often builds solutions to problems on top of enormous previous complexity. And in fact, in many ways, an analogy is a Rube Goldberg device. This is a device that puts together 14 different gadgets that allows Rube Goldberg to cool his soup. And if you think about this analytically and ask, how could you come to understand how such a complicated process worked? One, you'd want to get a complete parts list and understand what the individual parts might do. 2, you'd want to be able to connect those parts. 3, you'd want to be able to understand the dynamics of how the parts operated, eventually to cool the soup. And 4, you'd like to understand in the context of all of those properties the mechanistic explanation for soup cooling. And I would argue those elements are the essence of systems biology. With any complex system, it's exactly the same 5 features that you'll want to explore. And as was noted earlier, in 1973, I read this book you've already heard about, The Structure of Scientific Revolutions by Thomas Kuhn, and it had an enormously powerful impact on me. And the basic idea was there that new ideas struggle enormously to get by the intrinsic conservatism, particularly I think of scientists and physicians, but once they do, they really transform the field. And that actually got me thinking about this whole idea of complexity. And over the last 45 years or so in my career, as you've partly heard in the introduction, I did participate in I would say 5 paradigm changes that have integratively led to this transformation in healthcare that I'm going to be talking about. So one was bringing engineering to biology, and actually more recently we've developed another instrument that can do single molecule analysis in interesting ways. But these instruments allowed us to read and write DNA and proteins in various ways, and they created the opportunity for high throughput biology, which led to big data. And those are central features of contemporary medicine. And of course, the automated sequencer made it possible to do the project, the Genome Project. I got involved very early in the Genome Project. And again, it really harkened back to Thomas Kuhn, because I remember NIH in about 1985, 86 saying, we're already doing everything in the Genome Project. It's called genetics, and we're spending $300 million. And what's really interesting about new ideas is it's so easy for people to say, we're already doing that, and it's not true. They don't really understand what the differences are. The Genome Project, of course, gave us the parts list for humans, both specifying genes and by inference specifying the proteins, and the need to put together engineering, computer science, chemistry, and biology to create the automated sequencer led to the concept of this cross-disciplinary biology. And the idea, it's very common now, we should have all of these flavors of scientists together working in an integrated way on problems of enormous complexity. And Bill Gates made it possible to set this department up, and the department was remarkably successful in creating transforming tools for genomics and proteomics and cell biology. But what was quite clear is in bringing systems biology to reality, that the bureaucracies of science really hindered many of the new things that had to be done. And that was the reason why in 2000 I resigned from the university and started the Institute for Systems Biology and developed these ideas of system science over the next 14 years. And it was then that we started applying these systems approaches to disease that led to the final paradigm changes of systems medicine and P4 medicine. And we'll talk about those throughout the rest of the lecture. Now, if you ask me what are the really central features of systems medicine, I would say there are three. One is this idea that we now can create for each patient a data cloud that has billions of data points, and we're beginning to have the analytic capabilities for integrating and modeling these data for each individual to optimize wellness and minimize disease. And of course, in big data, there are a number of challenges. One is, how do you integrate together multi-scale data? An enormously challenging problem that I would say is one of the central challenges of contemporary systems biology and systems medicine. But equally important is the idea that in all big data, there are enormous noise to signal problems. How do you extract a very small amount of signal from an enormous amount of noise? And the idea that you need enormous domain expertise in biology is central to being able to do that. And I'll give you an illustration of that in just a few moments. Now, one of the ways you can reduce enormously the data dimensionality of these personalized data clouds, of course, is to convert them into what we call a network of networks. Networks in biology are processes that pick up information, modulate it, transmit it, and then give it off to machines that operate the functions of biology, execute the biological functions. So networks can operate at the chromosomal level, at the molecular level, at the level of cells, at the level of organs, and finally at the level of individuals themselves. And of course, these molecular networks are absolutely critical in many ways for understanding the essence of biological processes, be they health or be they disease. And of course, in disease, what happens is these networks become disease-perturbed, and that alters the amount of information that you can capture. Once you can assess how that information has been altered in disease-perturbed networks, you have fundamental insights into disease mechanisms, and you can use those insights to generate powerful new approaches, both for diagnostics and for therapy. So the third thing about systems medicine is how we view data. Our analyses should be as global as possible, because you never know which elements of information are involved in mechanistic processes. It's critical to do the dynamics of these systems, both temporal and spatial dynamics, and it's the dynamics that are one of the most powerful approaches to dealing with signal-to-noise kinds of problems. Integration of data, we've already talked a little bit about, and I'm going to show you in just a moment that noise really is made up of two different entities. One is the technical aspects that come from gathering the data and manipulating it, but much more difficult is biological noise. That is, when you measure any aspect of phenotype, normal biology feeds in as well as the disease biology. How do you subtract away the normal biology so you can come to understand what the essence of the elements that contribute to pathology really are? We'll talk about that in just a moment. So I'm going to talk about 4 of the 5 conceptual pillars in dealing with complexity in systems biology, just the simple view that biology is an informational science, and that is critical because it gives us a conceptual framework for dealing with complexity. I'll give you some simple examples. We've created a real infrastructure and culture for doing systems biology, and understanding what that is about is really central to being able to practice it effectively. The holistic, dynamical approach to studying systems is key. I'll give you one example there. And then I'm going to talk about emerging technologies and systems-driven strategies that really are transforming medicine and eventually will be transforming healthcare. So what about this idea of biology as an informational science? So the first concept is there are 2 fundamental types of information that are processed in living organisms. One is your digital genome, which gives you the reference set for your potential capacities. Let me just say, though, it's really important to say the genome does not determine your destiny. The second type of information, environmental information, modulates what the genome does, and you can take advantage of that very much to modulate your potential output, and we'll talk more about this later. So the genome is really critical, and that sets a framework for thinking about biological information. But understanding how the environment from the outside operates on that information is really one of the big challenges. These 2 types of information together create living organisms. That is, they give you the phenotype, be it a normal or diseased phenotype. And of course, we can ask the question, what connects these types of information with the phenotypes? And we would argue that there are 2 fundamental systems that do. One is the biological networks, which capture and transmit and modulate information. And of course, the other are the simple and complex molecular machines. And indeed, the dynamics of these 2 types of processes are absolutely central to understanding systems approaches to both biology and to medicine. Biology, as we all know, is an enormous hierarchy of information. DNA to RNA to proteins and metabolites and to networks and modules and all the way up to cells, organisms, and ultimately ecology. And the really important thing to understand is that every each one of those levels, the environment impinges upon and modifies the core digital signal from the genome. Hence, if you're to really understand a system, say at the level of the cell, how cell division works, what you would ideally like to have is the information at all the preceding hierarchical levels so that you could integrate it together and in doing so explicate the nature of the environmental impositions that have modified the digital signal. And this is what the essence of the integration of data types is really trying to do. Okay, let's talk about systems biology and the infrastructure and culture. When I started the Institute in 2000, the fundamental mantra I had was, as you heard, let's practice biology at the leading edge, but if you do so, very quickly you realize that you need to develop new technologies to explore new areas of data space. And if you develop those new technologies at the same time, you need the analytic tools to be able to handle and integrate and model all the information. And to do that, you needed to bring together all the scientists shown on the right-hand side of these slides, but bringing them together is utterly insufficient. They have to learn one another's languages. They have to be present in close proximity for lots of interactions, and they have to learn to work effectively together with teams. And it sounds like a lot of the things engineers are very familiar with. And we've taken 8 or 10 years to really learn how to do that well, and I think we've achieved that now. And if you do that, you can develop new technologies, you can develop new algorithmic formulations, you can develop new concepts of biology. So this is an engine that drives discovery, but it's also an engine that drives innovation. And using the principles here, in the last 14 years of ISB's existence, we've participated in starting 19 different companies in the Seattle area and other areas, and essentially all of them are still operant, and I think some of them will be very effective. But to give you another view of success, one of the metrics that we've been pleased with was the analysis by a Spanish institute of 3,300 research institutes a couple of years ago with regard to the impact of their papers. And this shows that ISB was 4th in the world in the impact of his papers, and our impactful papers were in biology, in medicine, in technology, and in analytics and mathematics. So it gives you a very powerful algorithm for changing the world. What about holistic dynamical systems approaches? We've studied now for 8 or 9 years the dynamic approach to neurodegeneration in mice that's induced by injecting into the brains of mice prion particles that have been activated to trigger the disease. And what we did in this disease, which progressed over a time course of 22 weeks or so, is look at 10 different time points along the progression of the disease at the brain transcriptome, that is all the messenger RNAs made in the brain. And we subtracted the brain transcriptome of normal animals from those of the diseased animals at each stage to be able to get the genes that were differentially expressed. And to our horror, we found that about 1/3 of the mouse genes were differentially expressed in the mouse brain. But what was obvious is most of that differentially expressed information was biological noise. So we constructed 6 inbred strain, prion strains of mice that subtracted away most of the normal sources of biological noise, and we ended up with 300 genes that probably encoded the neurodegenerative process. We also looked at the same time pathologically at the dynamics of this disease and showed that there were 4 major processes operating. something called prion replication accumulation, glial activation. It's an innate immune response, and two different forms of neurodegeneration. We had networks for each of those four processes, so we could feed in the information of transcript levels at those 10 different time points, so we could, for those four processes, look at the dynamics of their disease-perturbed networks. And two really important observations came out. One was the idea that these four major networks were sequentially disease-perturbed, starting initially with the most unique network for the disease, prion replication and accumulation, and then glial activation, and then the two forms of neurodegeneration. This is really key because it has enabled us to look at the earliest mechanisms of this disease process and come to understand in really deep ways basic initial disease mechanisms, giving us the tools we need for early diagnostics and for beginning to think about therapy. And we'll return to this theme at the very end of my lecture when we talk about how we can do exactly the same thing in human beings. What was interesting about the 300 genes we plugged into these four networks is only 200 genes fit into the networks. The remaining 100 defined 6 smaller networks no one had any idea were involved in this process. And that shows you how why global analyses are important. We never know a priori what's involved in a disease process. And what was interesting then is if we looked at the dynamics of each of those 10 different networks, they explained in toto virtually every aspect of the pathophysiology of this disease process. And I'll just say we've looked at a whole series of other animal model systems for neurodegeneration, for post-traumatic stress disorder, liver toxicity, and for cancer. And we have exactly the same dynamics we've been able to show for all of these, giving fundamental new kinds of insights. Let's talk about emerging technologies and systems-driven strategies, starting first with technologies that we're interested in at ISB. And I'm only going to talk about the first of these technologies, and I'll cover a couple of others a little bit later. We had a company from a spin-off from Intel come and take residence at ISB about 2 years ago. And they brought with them really a fantastic technology. Namely, they had learned to put together two different plastics in such a manner that they could create pores that had precisely controlled dimensions. And what was interesting about that, they proposed to use this technology to park a DNA nuclease on the lip of such a pore, to have it capture a strand, cleave off the individual bases, send them through the pore, and they've demonstrated that electronically you can distinguish the 4 bases as they migrate through such a pore. And of course, what is really interesting about nucleases is they are infinitely processive, so we can readily envision that this approach will give us very, very long sequence reads, 200 or 300 or 400 kilobases in length. What is also interesting is there are nucleases for RNA as well as for DNA, so this machine can sequence full-length transcripts beautifully for RNA, or will be able to. We haven't demonstrated that yet. But the other thing that's really interesting is if you can distinguish the nucleotides migrating through the pore, we have a real shot at being able to identify the 16 different epigenetic marks that can occur on DNA molecules by their electronic signature, methylation, and many other kinds of things. So you'll be able to get at least the covalently DNA-associated epigenetic marks at the same time you do the sequencing. And of course, the final idea is you can put these plastics together in such a way that you can infinitely parallelize the number of pores that you create. And we've done calculations that show in a 5 to 8 year period, we think, with proper funding, that we ought to be able to sequence a human genome in 15 minutes and do it for a price of $100 or so. So third generation sequencing is really going to be transformational. The strategies that we've developed at ISB for looking at disease processes include family genome sequencing, which integrates genetics and genomics to define disease genes. It includes a systems-driven approach to blood diagnostics. Let me say that the difficulty with blood diagnostics is there are lots of differences between normal individuals and disease counterparts, and 99.9% of those differences are noise, and you have to understand how to get the signal out of those things. And I'll give you an example of a lung cancer diagnostic that we've developed. And then I'll talk about a futuristic global assay, mostly to tell you about a new kind of protein capture agent that we're developing. So, integrating genetics and in genomics, we started in 2009 with a family of 4. The kids had 2 genetic diseases. The parents were unaffected. We sequenced all 4 genomes and got all sorts of information. We could correct more than 70% of the sequencing errors. We could identify rare variants as opposed to signal if 2 or more members of the family had them. And we could ascertain the blocks of related chromosomal segments that were passed from parents to the kids, the so-called haplotypes. And those are really important for reducing enormously the chromosomal dimensionality within which you have to search for disease genes. And we identified 4 genes that were the likely candidates for these 2 diseases, and the assignment of correct genes was very simple. We've developed an enormously effective platform for doing family genome sequencing and using all the power of Mendelian principles, and we'll talk about how we're going to use this platform to do interesting things toward the end of the lecture again. And I'll give you an example of a childhood neurodegenerative disease where we looked at 5 families. The top family here had a gene that had already been defined for this disease, but it came out of looking just at that single family. When we looked at 4 additional families, we identified 3 genes that had never been seen before, and again, each disease gene was identified from analyzing just 1 family. It obviously had to be confirmed by other kinds of studies, but this is the power of what we've done here. And I'll just say parenthetically, recently, we've looked at bipolar disease, studied 43 families with multiple affecteds against 34 control families, and we've identified a cadre of disease-causing genes that all have to do with neuroexcitatory events, and in all cases, the diseased individuals has 2 to 3 or more of these disease genes as a part of the whole disease process, though this gives us a completely new approach to the stratification of disease genes. So over the last 4 or 5 years, we at ISB initially had generated about 1,700 complete human genomes, and more recently, in collaboration with Innova, have added another 4,000 plus genomes. So we now have 6,000 complete human genomes, and what this allows us to do is to carry out with those 6,000 genomes preliminary analyses to make a reference model that uses at least 6 or 8 different tricks to increase the accuracy of our ability to interpret new individual genomes that come into the process. And that's, again, really critical for this wellness study I'm going to be talking about later. What about this idea of a systems approach to blood diagnostics? There was an OMIC report a couple of years ago that looked at all the preceding blood diagnostics that had been done, and they concluded that essentially none of them had been translated to clinical practice. And yet you have Science, Nature, Science Translational Medicine papers and everything. And they said the faults were twofold. One is this idea I've already told you, look at blood, normals and diseased, and lots of differences, but most of them are noise, and most people don't know how to deal with noise. But the other point is, if you look at an individual population, the genetic polymorphism can be confounding because you can get really good sensitivity and specificity for that one population that doesn't work on other populations. So when you do your diagnostic markers, you should be doing them in the context of 4 or 5 different populations at the very beginning, so you weed out the enormous noise that's associated with polymorphisms. Blood is an ideal diagnostic vehicle because it bathes all organs. They secrete molecules into the blood. Those molecules, as we'll see later, can be enormously informative with regard to the origin of changes that are occurring in the biological system. Longitudinal analyses are key. Multi-parameter analyses are key because you want to sample multiple disease-perturbed networks to create these diagnostic panels. Quantitation is critical. And I think proteins are going to be the best biomarkers by far because they're closer to biological activity. Nucleic acids really have significant signal-to-noise problems and so forth. And of course, the tool that we're using now to look at proteins is targeted mass spec-based proteomics, where we have the ability to look at 100 or 200 proteins and to quantify them within an hour or so using heavy isotope peptides. And I won't go into the details. One thing that Bob Moritz at ISB has done is develop these targeted proteomic assays for virtually all known human proteins. So we have lookup catalogs that can create assays virtually simultaneously for a whole panel of new proteins that we'd like to explore. We've actually used this kind of approach to look at this Prion-based disease that I talked about earlier through the blood. And one of the pioneering concepts we've introduced is the idea of organ-specific blood proteins. The idea is we've analyzed 50 transcripts. We've analyzed transcripts from 50 different organs in both mouse and human, comparing each transcript against all others to ask which transcripts are relatively organ-specific. Then we've asked, do those organ-specific transcripts make a protein that's secreted into the blood at levels that we can detect it with targeted proteomics? And we have about 200 proteins that map beautifully into networks that encode all of the major biological functions in the brain. And of course, the idea is for a normal brain, the level of those proteins will be set at one series of concentrations. And in a disease brain, those blood-specific proteins whose cognate networks have become disease-perturbed will change their concentration level. And because every disease has different combinations of disease-perturbed networks, you can uniquely separate well from disease, and if disease, which disease it is, if you have the right patterns to be able to do the diagnosis. So in prion-induced bloods, bloods from animals that had prion-induced disease, we did a longitudinal study from beginning to end, looking at 15 brain-specific blood proteins that map pretty evenly into the 4 major networks that I described earlier, and we were able to demonstrate several really important things. One, we could detect the changes in this disease almost simultaneously with when we saw the first disease-perturbed network, so very early diagnosis. And number 2, we could see in the blood the sequential disease perturbation of each of the 4 networks. Hence, we could follow the progression of the disease. And keep that in mind as we talk about human disease now in a a lung cancer blood diagnostic panel that we did with this company I started about 5 years ago, Integrated Diagnostics. And the basic idea here is, in the US, there are 3 million lung nodules that are detected by CT scan a year, and what the pulmonary oncologist faces is, what do we do with these spots in the lung? And it turns out, of the 3 million, about 600,000 fall in a dilemma zone where they're uncertain about watchful waiting or immediate surgical resection. And often what happens with most of those is they are operated on using various surgical procedures. And the tragedy is that about 60% of those operations are done on benign nodules. Hence, it's costing the healthcare system money, and it's causing the patients a lot of morbidity. So our idea is to set a panel up that can distinguish benign from neoplastic nodules. And the approach that we took was to use systems thinking to identify a large panel of 370-some candidate proteins. And we actually got them a number of different ways I won't go into, but we were able to only reliably detect about a half of these 190 in the blood. So we then moved to analyzing the 190 with regard to 144 plasmas, 72 from individuals that had proven benign nodules, 72 from individuals with proven neoplastic nodules. And what we did was rank order the 190 proteins with regard to their ability to make those 144 calls. We then took the top 32 out of the 190, there was a break point, and we did the following. We asked a computer to generate 1 million panels of 10 randomly selected from those 32. Then we rank ordered those panels with regard to their ability to, again, make those 144 calls. And we found that we then looked at the top panels of 10 and asked the question, which are the proteins that are found most frequently in those top panels? And we called those cooperative proteins. And it turned out that we were able to select 13 proteins that were repeatedly found in those top, and those became the panel then that we did a validation study on. And in the initial study, we used 4 different sites. In the validation study, we used 5 sites. We brought in a new site and did a similar kind of analysis, and it passed with flying colors. And so we then commercialized in the 4th quarter of last year this panel of 13 proteins to make that diagnosis. And the acceptance of this panel has been remarkably quick. And the reason for that is we could rule out about 40% of the benign nodules with a 90% specificity, cutting out more than 1/3 of the unnecessary surgeries. And that's alone would save the healthcare system about $3.5 billion a year. And that was the fact that really persuaded the payers to adopt this study, in spite of the fact it's also going to improve healthcare as well. Now, we did all this systems filtering, so a really interesting question is, what do these 13 proteins represent biologically? And interestingly enough, 12 of the 13 map into one of 3 disease-perturbed networks that play a major role in lung cancer. So this panel will also give us the ability to follow the progression of lung cancer and do other interesting things as well. Without going into any detail at all, I can say we've taken a similar approach to post-traumatic stress disorder, and we have beautiful panels now that have close to 95% sensitivity and specificity for distinguishing normal soldiers from Afghanistan from those that have severe post-traumatic stress disorder. So this is the first time ever that we've converted a disease that heretofore had been diagnosed psychologically into a quantitative blood test. And I think we'll be able to do this for all neuropsychiatric diseases and fundamentally change how we approach those diseases in the future. So these panels can do 7 things: distinguish normal from disease, carry out early diagnosis, follow the progression of the disease. They will be able to follow beautifully the response to therapy, obviously can follow the recurrence of disease in diseases like cancer, and they'll point us toward the key disease-perturbed networks which themselves pose candidates for drug target identification. And we think these panels will let us stratify most diseases into their discrete subtypes, each different subtype employing a different combination of disease-perturbed networks. So let's talk about a wellness assay that I hope in the future can be done at home with a fraction of a droplet of blood in a few minutes, and we'll be able to analyze 2,500 organ-specific proteins using ELISA assays with peptide protein capture agents. This is a collaboration we've been carrying out with Jim Heath at Caltech, and he already has set up a microfluidic platform that can do 50 assays using commercial tests that exist. And the idea is if you could get 50 organ-specific proteins from each of your 50 organs and carry out this test 3 times a year, you'd be able to identify instantly subtle transitions from wellness to disease and begin to do things immediately. That's one of the hopes for the future. So this assay Jim has set up uses a fraction of a droplet of blood. It's done in 5 minutes. It's mid-attomole level sensitivity at least, and it's already been used in a hospital. What is really critical in doing this is how do we get 2,500 ELISA assays? That would probably cost, if you used monoclonal antibodies, several $billion at a minimum, an impossibility. So Jim has really pioneered the development of peptide protein capture agents that we think will totally replace monoclonal antibodies. And the essence of the idea is he's created libraries of peptide 5-mers that are circular, D-amino acid, can't be cleaved by L-amino acid enzymes. And you can take a protein and you can use a library that has 10 to the 6th or 10 to the 7th such circular proteins to identify low affinity binding circular peptides. Then you can employ a chemistry called click chemistry, which can actually join together 2 of those circular peptides in precisely the correct 3-dimensional orientation to make a dimer or to make a trimer of these reagents. Dimers usually give us an affinity that approaches some of the best monoclonal antibody affinities. What's great about these reagents is they're rock stable. You can put them at 60 degrees for months. Their affinity is unchanged. What's exciting is you can put these capture agents in an envelope and mail them off to Africa, and they'll still be completely functional. Not true of monoclonal antibodies. One of the exciting recent events is we've used key peptides at epitope sites and proteins we'd like to target the protein capture agents to demonstrate we can do just that with such precision that we eliminate virtually all of the cross-reactivities that plague monoclonal antibodies. And so we can get reagents that are precisely targeted just exactly where you'd like them to be targeted. And we've actually used one of the reagents directed against human PSA labeled with copper-64 to show with PET scanning that when injected into a mouse, this reagent localizes relatively quickly and totally specifically in the prostate. And of course, what this gives us is the beginning of the way to look at molecular dynamics in a spatial sense, and that's one of the key features of systems biology. So these circular peptides that we use to create these dimer reagents are stable. They're highly sensitive. Each mer you add gives us at least a log increase in sensitivity, so we could even go as sensitive as one would want. They're digital in the sense that once you know the two circular peptides plus the nature of the click chemistry linkers, you can do everything in vitro with copper catalysis. So you can generate enormous quantities, and you'll forever have the formula for creating these protein capture agents. They're precise, they can be directed epitopes and avoid cross-reactivity. And we recently created this company called Indie Molecular that's going to adapt this approach to very large-scale production. So you can do in vivo, in vitro diagnosis. I'm most excited about the possibility that we can create therapeutic reagents from these things. And we've already shown with epitope targeting that we can inhibit the activity of key proteins either allosterically or by direct blocking kinds of functions. So we feel that this is the approach we'll take in 5 years or so to have the 2,500 organ-specific ELISA assays. And my prediction is these will replace monoclonal antibodies in the next 10 to 15 years or so. So, systems medicine has reached this enormous tipping point, and you can see we can do dynamics to get molecular mechanisms. We can do family genome sequencing to identify disease genes. We can get panels of diagnostic markers to distinguish various aspects of disease. I haven't talked about it, but we can stratify disease. We can stratify patients. We've shown recently, for example, in 100 patients with Alzheimer's disease, using liver-specific blood proteins, that their liver changes really quite profoundly. So most diseases aren't going to be single organ, they're going to be multi-organ, and these open up new diagnostic and therapeutic possibilities. We can look at disease-perturbed networks and develop computational tools to identify in a new way candidate drug targets. What pharma's great at is making drugs. What they're terrible at is choosing the targets for the drugs. And this opens up really powerful new approaches for doing just that. And I'll talk about one longitudinal study in just a few moments. So I would argue that P4 medicine thus arises from a convergence of systems medicine on the one hand, from the big data and the digital revolution on the other hand, and on the third hand, the importance of consumer or patient-driven social networks. Why I think they are so important is because the healthcare system and physicians are enormously conservative. Driving through change is unbelievably difficult. The best agents for driving change are patients, and they can work marvelously effectively. If they go to their physician and say, you don't know anything about genomics, you don't know anything about nutrition, you don't know anything about inflammation, I need a doctor who does. That's motivating, or it's time for them to retire if they can't be motivated. Now, P4 medicine differs from contemporary medicine in 6 different ways. 1, it is proactive. 2, it's focused on the individual. 3, it's focused on wellness. 4, it's all about generating these patient data clouds that will allow us to produce actionable changes in the individual's life, optimizing wellness, minimizing disease. And of course, it really looks at clinical trials in a very different way than contemporary medicine does. So if you wanted to do a cancer study, you might take 30,000 patients, you might give them cancer drug or placebo, you'd extract the results into a series of curves. From those curves, you could make predictions both about how the average population behaved and about how good the drug was. And I would say that approach is flawed in every way, because every single one of those 30,000 patients is unique genetically, and they're unique environmentally, and you can't do statistical averages of different kinds of entities. So what What P4 medicine does is to analyze each individual in the context of themselves, their data cloud, and after you have 30,000 of those, you can aggregate the individuals based on their own unique property according to those that will respond to a given drug or whatever feature you might want to carry out. And I'm going to talk about N-of-1 experiments in a few moments, which most physicians hate because of the lack of statistical power. And I think N-of-1 experiments are really going to be the future of medicine in really interesting ways. And of course, the patient-driven social networks we've already talked about. So this P4 medicine then is about two things, wellness and disease. And of course, contemporary medicine is 98% about disease and very little about wellness. And in fact, if you think about contemporary medicine, I've always thought it's very much like the fellow looking for his coins lost 2 blocks away under the lamppost because there's light there. So the questions I'd ask is, wellness is a really important concept. We have no way to quantify wellness, really a key point. Number 2, we have no way to analyze disease in humans at the earliest stage. In fact, we only study people after they get sick. And many doctors will say, you're well, come back to me when you get sick if you have abnormal chemistries. So how do we look at the beginning stages, and how do we look at the entire progression of the disease from beginning to end? And that's what I think we can do with this P4 pilot project, which is a digital age study, longitudinal study of 100,000 patients over an extended period of time. So what we will measure from the blood is your complete genome sequence. We do that only once. And then all the other measurements I'm talking about will be collected every 3 months. We're going to do clinical chemistries. We're looking at 120 metabolites, 15% of which come from the gut microbiome. And we're looking at organ-specific proteins for heart, brain, and liver to detect early disease transitions. From the urine, we're interested in oxidative stress and nutrients and cellular energy metabolism and so forth. From saliva, we're looking at markers of stress. From the stool, we're looking at the gut microbiome to quantitate diversity and the nature of the populations of bacteria that are there. We're using quantized cell self-tracking devices like the Fitbit for activity and sleep, but weight isn't critical as are blood pressure and heart rate. And then we're having patients keep track of their personal health status, both physiologically and psychologically. So if you think about it, if we start with 100,000 individuals, in a period of time, they'll separate into 2 distinct categories. 1, those that remain well or improve in health, and 2, those that transition into disease. And once we're able to detect those 2 distinct groups, we can do really interesting things. For example, we can analyze these enormous data clouds of the well patients to get multi-parameter metrics of wellness, which I'm utterly convinced are going to define some of the most basic psychological determinants of what it is to be well, just as we did with PTSD and measured stress, presumably there. And I can't stress how important these multi-parameter wellness panels are really going to be. On the disease transitions, we'll have the ability to go back, because we've got these data sets every 3 months, and look at the very origin of that disease transition. Come to understand, as we talked about earlier, disease-perturbed networks, the basic mechanisms, and get then early diagnostic tools, as well as the drugs that will allow us to transition the patient from this disease back to a wellness trajectory and save all the downstream disease dollars that we've avoided. So the idea is, with all these assays we've discussed, we'll integrate them together for each individual. We'll create a model for each individual which delineates all of the actionable possibilities that individual can use to improve health and avoid disease. We've actually started in March, 5 months ago, with the first 108 individuals on this project. We're just gathering the second 3-month set of data now, and the analyses of these data have absolutely been spectacular. One, we've determined that there are an enormous number of actionable traits. And an example of an actionable trait, a friend of mine at Microsoft 13 years ago was told he had irreversible osteoporosis. He was going to be in a wheelchair the rest of his life. He had to give up working and everything. He had a genetic analysis done that demonstrated a defect in a major calcium transporter. And on his own, he started taking 20 times the normal amount of calcium, and in a year and a half had reconstituted his bone structure, and 13 years later on the same regimen is perfectly healthy and still functioning as he did before. So that's an example of an actionable kind of trait. And I'd argue from the individual data types, we'll see many actionable traits, but especially when we begin integrating different data types, we'll reveal new kinds of actionable traits. And I'll give you an example of one. In this 100 Pioneer study we did, we were really shocked to learn that 90 out of 108 individuals had really low vitamin D. And vitamin D is associated with a lot of really bad things. But it also turns out that there are at least 3 genetic variants in your genome, each of which has the ability to significantly block the absorption of vitamin D. So if you have one or more of those defects, it almost certainly is going to take 20 or 30 times the normal amount of vitamin D to bring you up to normal. But if you didn't have those defects, if we gave you 20 or 30 times the amount of vitamin D, we could throw you into vitamin D toxicity. So you need to know the genome, and you need to know what your vitamin D levels are. And that's the integration of data types that let us get actionable possibilities. We've recruited coaches that take these actionable possibilities with MD advisors and bring them to the patients, both to explain the opportunities and to convince them to act upon the opportunities. And this has worked incredibly well. And finally, we're creating social networks. And of course, they're useful for learning, they're useful for crowdsourcing, and they're going to be useful in the future for advocacy. So in the 100 Pioneer Project, cardiovascular patterns, there were 57% who had actionable possibilities there. Pre-diabetes, we found 32% were affected. We, in fact, found one individual that had frank diabetes, and it was early enough to be able to reverse it now, which is wonderful. Inflammatory patterns, nutrient insufficiencies. It is amazing how many well people have major nutrient insufficiencies, and this gives us wonderful experiments to do on this N of 1 category. So for example, with vitamin D deficiency patients, we're putting together genetic and the blood quantification of vitamin D to create regimens to see how much it takes for each of the 3 different kinds of variants to be able to bring your vitamin D levels up to a reasonable category. 100% of the pioneers have 2 or more actionable traits, and we haven't done much data integration so far, and we're just beginning to get some of the final data types. So again, your genes don't provide your destiny, they provide your potential. You can avoid a lot of potential consequences or enhance them if it has to do with wellness by your own behavior and activity. So at the end of this year, we'll start on 1,000 individuals. At the end of another year, 10,000, and then finally move to the 100,000 Wellness Project. We're collaborating with NIST to set standards for both data types and sample collections. We've had individuals from 15 countries contact us about helping them initiate their own 100K projects. Enormous enthusiasm for this idea. I'm considering taking this and making it a second genome project, in the sense that we'll go to Congress and argue, as we did for the genome project, that there are compelling opportunities for improving health, reducing health costs, promoting innovation, providing a leadership role in medicine for the US. And I think the arguments are better, frankly, than they were for the genome project. We're going to pursue two strategies going forward. One is this large 100,000 person discovery project, and it's really important to study the general features of what we need to do. But at the same time, we've just started a wellness company, and it's here I think we can really scale this whole effort to think about exporting it initially to the developed world and ultimately to the underdeveloped world. And I think this has the potential to lead to a democratization of healthcare, which will be transformational. Moore's Law is going to decrease the cost of all of these assays. They are currently about $13,000 per person per year, and I think in 5 years they'll be less than 5% of that. DNA sequence costs are one example of that, and there is an enormous opportunity for driving new technologies from machines to smartphones that people can use at home. The opportunities, I think, are legend. We'll have a catalog of actionable possibilities. We'll be able to look at each individual and optimize their wellness and minimize their disease. We'll create these multi-parameter wellness metrics. My feeling is wellness is like a deep well, and most of us are at the bottom of that well. And with the right guidance, you can optimize your properties, get to the top of the well, and give yourself more energy and excitement and all the kinds of things that are good for life. So defining wellness is really critical. We'll look at early disease transition mechanisms and begin in time to divert people from being sick back to being well very early. And I think the ability to drive the development of new assays is an enormously exciting kind of opportunity for the future, and it'll be interesting to see in that regard. I'll bet a lot of the things we hear this afternoon will be very relevant to that. This, if you think about it, is P4 medicine. We'll be using it to bring it into the healthcare system. So what about society in general? How is P4 medicine and the 100K project going to impact that? One, it's going to lead to a digitalization of medicine for the individual patient. And digitalization always means it's cheaper and means it's exportable to developed as well as underdeveloped countries. Two, we're going to turn around the costs, ever-escalating costs of healthcare. I don't think the contemporary healthcare system can ever turn those costs around, because I think a really key part of turning the costs around is making the patients take the individual responsibility for their wellness. And our 100 pioneers are doing this, and it's really been transformational. We're going to improve the quality of healthcare. We're also going to force all sectors of the healthcare industry to revise their business plan in accordance with the dictates of P4 medicine. And I think many aren't going to be able to do it, and they'll open up wonderful ecological niches for new companies that are fashioned to the dictates and opportunities of P4 medicine. And finally, I'd argue that there will emerge over the next 10 to 15 years a wellness industry that far exceeds in market cap our current healthcare industry, and that we're today creating the companies that will be the Googles and Microsofts of the wellness industry, and enormous economic opportunity for organizations that are willing to adopt the principles of P4 medicine. Lots of people have been involved in this, and I won't list their names, but thank you indeed for your patience and attention.
So on behalf of the 2014 EMBC conference and the organizers, as well as all of you who participated in planning the meeting, Dr. Hood, we have a small gift in token of our appreciation for your talk today. Thank you very much.